Thing. There we go. All right, everyone. Thank you all for joining tonight on today, Tuesday, August 26th, 2025, for the meeting of the Somerville Biosafety Committee. We're meeting today to discuss a single item, which we'll dive into here shortly. But first, a little bit of formalities. Pursuant to Chapter 2 of the Acts of 2025, this public meeting will be conducted via remote participation. A recording of this meeting will be made available along with the minutes on our website, somervillema.gov/biosafety. I just want to thank everyone for joining tonight. We don't have a large agenda. We have a single presentation for review. Committee members have been provided the application along with all the supporting documents for their review. So we'll ask questions at the end of the presentation. If you do have a question, comment, or concern, please use the raise hand function at the bottom of your Zoom web page, and you'll be recognized for your time. To start off, I'm going to conduct the roll call to start. All right. Elise Ballarge. Not here. Rachel Nadkarni. I see Rachel. Rachel, are you here? Alex Vera. Deputy Chief Bill Hallahan.
I am here.
Francine Rogers.
Present.
Michael Swickus.
Present. Present.
Bill Fisher.
Present.
Excellent. Karen Carroll.
Present.
Present. And I'll wait for Rachel. I'll mark her as present if she responds. But oh, there she is. Hi, Rachel.
Present.
Oh, she's there. All right. Rachel is also present. With that, we have a quorum. So I'd like to start off with our first item, which is the presentation of applicants for review and discussion, specifically Empress Therapeutics, which is seeking to operate at 101 South Street here in Somerville. Do I see a second? All right, seeing a second, we're going to move into our presentations. So just for clarification, who from Empress will be handling the presentation and would be like to be made presenter?
Well, I mean, I can drive or Sarah, do you want to drive?
Do you have the presentation handy, Dawn?
I do, yeah. So I can present, sure. And then you just tell me when you want to advance. Yep.
Sure.
And so it'll be a mix of Sarah, myself, and Lewis, who's the operations manager, presenting. And so I will share my screen.
Excellent. Don, you've been made co-host. The floor is yours, okay?
Awesome. All right. Go. Let's get that into, there we go. Okay. Get in presentation mode. Here we go. All right. So I want to kick it off by thanking everybody for their time this evening. You know, I know it's summertime and people have a lot of interesting vacations, including Scotland. So thank you very much for taking this time out of your schedule to consider our application. So we'll start with a company overview. So I am the leadership team at this point, right? So I lead platform development. We're a platform company, and you'll learn a little bit more about what our company does. We'll also have Lewis cover the actual physical space, and the safety paths and all that. And then we have Beth Culp is a scientist serves as our biosafety officer. And then Xiaowei Guo is our senior scientist, and he serves as our chemical hygiene officer. So we're good to go here. Right, we'll advance here. So we're founded in 2018. Well, operations plan to start in Somerville. Well, actually, we've already moved to Somerville, and so we've been setting up the lab, but haven't really uh, conducted any experiments that are covered under this application. But we hope to begin that as soon as possible in September. We recently moved from Watertown to Somerville. We're currently 11 employees, and we occupy lab space on the 6th floor of 101 South Street. And we're a subtenant of Celerity Therapeutics, which is a company that's the main leaser of the floor. So you'll see that one of the permits is actually held by them in terms of the MWRA permit. So what do we do? So we develop novel therapeutics based on drug-like human experience molecules. So the microbiome, most of you have heard about that. The microbiome is a ecosystem of microorganisms that live within us. And the more we learn about it, the more we understand they're actually part of us. And if everything's going well, they are there to keep us healthy, and they do that by making small molecules that interact with our physiology to keep us healthy. So the main business model of Empress is to identify what those molecules are that are keeping us healthy relative to people with certain diseases, like inflammatory bowel disease. And then the idea is we're going to discover replacement therapies. So we find things that are overrepresented in healthy people that are missing in the disease state. And the idea is we'd make molecules that could help cure the symptoms that people by restoring them to the healthy state, right? So our platform uses artificial intelligence to identify the small molecules that are made by the microbes. And and then it's also based on principles of evolution, right? So the microbiome has evolved with us to keep us healthy, and we provide a lot of nutrient, important things to them like nutrients, right? So it's actually in their best it's in their best interest to keep us healthy, right? And so and over the course of many eons, there's a lot of different bacteria that make molecules that keep us healthy, right? And so we identify these molecules, and then we use various other methodologies to understand whether the molecules we've identified actually interact with human physiology, right? Sometimes the molecules that we identify made by one microbe will interact with another microbe, and then that microbe produces the thing that makes us healthy. So we really need the artificial intelligence to deconvolve a lot of those complex interactions. But in the end, we just want a small molecule that you can hopefully take as a pill, right? That can help ameliorate disease. So we identify the molecules, and then we need to be able to make those molecules, right? So that we can test them in human models. And we do that by a discipline called synthetic biology, where we identify what the DNA sequences are that encode the enzymes that make the molecules. And then we take those DNA sequences and we put them into bacterial hosts that we can work with in the lab. And then we identify what they're making, right? So we put them into a host that is otherwise clean, with only the genes that we care about are in there. And then we identify what molecule they're making, and then we isolate that molecule. And then once we have the isolated molecule, we're able to test it in human cell lines and things like that, right? So that's where our human work comes in. So we have a mix of bacterial work that has recombinant DNA moved into them, so they make the molecules. And then we have a mix of human work that we use lentiviral technology to actually identify what the targets are of the chemistry, right? So if we find chemistry that works in the cellular model, we actually want to understand what it's binding or interacting with. And that's where we use the lentiviral work to create constructs that can help us identify what the targets are of the small molecules, right? Okay. Okay, so this is a little bit more. So we culture the microorganisms. there's really common laboratory organisms, but we also use ones that, but there are strains that could be potentially be pathogenic. But all of those are classified as BL2. And this is where we insert the clone genes, so that the genes that we identify in the microbes, we're going to insert them into the heterologous bacterial hosts, and then we look at, what's different about what chemistry they're making. And then we just investigate the therapeutic potentials by a variety of computational tools. They're AI-based tools. When we treat the human or mammalian cell models with these molecules, we look to see what signature of treatment there are, and that signature of treatment is done by what we call multi-omics technologies. We're looking at changes in proteins, changes in RNA levels, and changes in metabolites, right? And all of that is used to figure out what the most likely targets are, and then we use the lentiviral technology that's detailed in our application to identify what the targets are. And that is what we call functional genomics, right? We identify what the targets are by interfering with the function of those targets using functional genomics. This is where the lentiviral work comes. We call it perturbation. And so we decrease the expression of the putative targets, and we ask, does it explain the effect of treating the cells with the small molecules? And that helps us identify that. We identify proteins bound to the small molecules with a methodology called chemoproteomics, which is we treat cells with the small molecules, and then we lyse the cells, we bust them open, and then we can identify what proteins that our chemistry is bound to by, excuse me, a methodology that's based on mass spectrometry, right? So we do a mix of nucleic acid genomics and also mass spectrometry to identify protein levels and also metabolite levels, right? And then once we know the targets, then these small molecules then become candidates for therapeutics, right? Because in order to finish your preclinical and then advance to clinical studies, you really need to know how these molecules function, right? And that's where we're looking at the signatures of molecule treatment in the cells, but also really what the direct targets are, right? And that meets the minimum bar for advancing in clinical studies towards a therapeutic that we hope helps lots of patients, right? Because a lot of the indications we work in, a lot of the standard of care doesn't work very well. So, for example, inflammatory bowel disease, the existing standard of care only works about in 30% of patients. So we're hoping that our small molecules will really close that gap. Okay. All right. All right. So, Louis, you want to handle those facilities part? And I can advance
Yeah,
slides?
I can take over for this. So,
Great.
the Somerville Empress facility is located on the sixth floor of 101 South Street, located in Somerville, Massachusetts. As Don mentioned earlier, we are subtenants on that floor. Solarity is the leaseholder for the sixth floor, and we are tenants of them in the lab and office space. The floor, the office and lab space requires badge access 24/7. The building itself also requires badge access 24/7. So that is to get into the building, also to get up to our floor. Key card access is necessary to get into the Empress space as well. This includes the lab space. Lab access is for authorized, trained employees only. So employees have to be trained by a safety consultant, whether that be Sarah, Anne Marie, and myself in attendance as well. And visitors that are visiting for Empress, they must check in with security. That is documented, and an Empress host has to go downstairs and let that person in. The visitors are always escorted throughout the premises. They're not allowed on site at any times without an Empress employee around. And while they're here, they must be escorted through different access points because everything is badge access, so they need to be accompanied by an Empress employee to get to those badge access points.
Great. Do you want me to go next slide?
Yes, next slide, please. And here we have a map of the bio waste transport. So that room that the arrow starts at on the left side of the screen, that is the biohazardous waste accumulation area. Basically, all of our sharp biohazardous waste and medical biohazardous waste is collected in that room. Then every Wednesday, our waste hauler, Stericycle, comes upstairs using the elevator, hauls the waste on that path that you see. That the red arrow, the destination of that red arrow goes to the elevator, which goes straight down to the first floor, and the Stericycle employee then hauls all of that waste directly into the truck. So they come up, grab the waste directly, and then bring it directly onto the truck from the room. Next slide, please. The Is this me?
No, it can be you or me, whatever you want.
You can take it from here.
I can take it from here. So, like I said, we have both bacterial strains, and also we use lentiviral vectors, third generation vectors. So they fall into the BSL-2. We have a separate tissue culture room to do all of the lentiviral work. That's within the badged lab space and with clear signage that indicates not only is it BSL-2, but that there's lentiviral work that going on in that room. Similarly, we have areas where the bacteria that fall under BSL-2, we have hoods that are not in the TC room because nobody wants microbes in their TC room. But we have a separate hood for that, clearly marked for BSL-2. We have a biosafety program in place, an exposure control plan, and these are reviewed annually by our we, as part of our operations function, we have, with our biosafety officers, we review these annually, and we communicate out to the rest of our company and new people are given the documentation and as part of the training, come up to speed on all the biosafety and exposure control plans, also any emergency safety plans we have. Okay, so we covered this. Safety training is conducted for all new hires. Currently, we do this in collaboration with safety partners, and those folks are on the call. Empress is contracted with Mount Auburn Operational Health Medical Services for post-exposure response. Hep B vaccination is offered to all lab free of charge. And like I said, Empress has an active safety committee that we meet once every quarter formally, but as part of our operations function that meets more regularly. Any incidences or things that come up, we get reviewed, and then we communicate back out to the rest of the company. We also communicate directly with the folks at Celerity, who also handle all of these things. So we have open communication with the folks that we share that lab space with. Okay, here's our list of materials that fall under the application. So mostly we work with the non-pathogenic E. coli, things like E. coli and Bacillus subtilis. Rarely, but we do work with these. We work with the organisms listed here that fall under risk group 2. We have human cell lines that we get from vendors. So they're tested for all the basic mycoplasma, all of that kind of thing. But since they may be engineered with lentivirus, we treat them all like risk group 2. And same with any human blood products we get. We typically get also from vendors some human blood products to understand the functioning of our molecules. And again, we use third generation lentivirus to do some of the target discovery work that's covered in our rDNA application. All that work is, like I said, done appropriately rated biosafety cabinets and labeled. So we have them all certified, and especially since we just moved, they've been recertified on an advanced schedule, so we're all good to go there. And the IB, we're in the process of scheduling our IBC, right? So that presentation is finished. We're just finishing up, lining up the folks that will participate in there. And then all disinfectant and practices and disposal methods will be reviewed and approved by the IBC. Okay. So these are our standard practices for liquids, media waste and aspiration facts. We just, pretty standard, decontaminated with 10% bleach for 30-minute contact time, followed by disposal in the sink. Any dry and solid contaminated things will be collected and packaged through approved containers, shipped through licensed Stericycle is our vendor that handles this. And similarly, any sharps, pastor pipettes, needles, scalpels. We have standard red, sharp spins that we also use Stericycle to manage for us. Okay. And so this is the status for all of our rest of our permitting platform, permitting process, RDNA, that's why we're all here tonight. Flammables, that's in progress. I mean, Sarah from Safety Partners has submitted the application to schedule the inspection. Likewise, the EPA ID application has been submitted, and we hope to get that soon because it was done several weeks ago. And like I said, alluded to before, the wastewater, the MWRA, is held by Celerity, our landlord. Here are the rest of our vendors. So we use Stericycle for biological waste sharps disposal. Hazardous waste is Veolia. Lab Coat Services Cintas, and Mount Auburn is our occupational health. We have all the fume hoods and both chemical and biological are certified by Air Systems. That's all complete. Pest control services, that falls under the landlord. That's complete. And our safety consultants are safety partners, and they're here today. Okay, and that concludes our presentation. We'd be happy to answer any questions.
Thank you, Don and Lewis. Really appreciate the presentation. Extremely thorough. I would like to open it up to committee members. If you have any questions, comments, concerns. Oh, Francine, I see your hand. The hand emoji thing is not popping up.
I think it blends in with my sky.
Okay.
I just, because I'm picky. One clarification, you did list on your slides that you're using Clostridium, but that was renamed in 2016. It was given to a different genus, so you might want to consider making sure that that's updated so that if people are searching for it, you're actually coming up with the Clostridioides and not Clostridium for your genus.
Excellent, thanks. As I'm an evolutionary genomics person by training, so I appreciate the correct phylogeny is important for sure. So yeah, we'll definitely update that.
All right, thank you.
Thank you, Francine. And Don, any updates you have to your application, any information, feel free to submit those back through the portal or directly to me to upload to the license approval. Okay. Sure. Great. I see Bill. Go ahead.
Thanks, Colin. I just have a couple questions about the emergency action plan. The first one is that when I look at it, you have procedures for emergency evacuations and partial activations. Later on in the procedure, you list shelter in place, but I could have missed it. I didn't see a procedure for shelter in place defined. Do you have that?
That's an excellent question, right?
And by the way, I write these all the time, and I end up on the other side of these questions all the time.
Yeah.
So, no worries.
No worries. Yeah. So not in the news space, right? So we can rectify that. Yeah, and make that more clear. Yeah.
Yeah, absolutely. Great.
Yeah.
The next thing is you guys do a good job in Appendix 3 talking about evacuations for persons with disabilities, but you list that there's no current employees that have disabilities. Just a
Mhm.
friendly suggestion, you guys, could have someone that has a disability that doesn't disclose it. You could
Yeah.
also have a temporary disability like an ankle or something, or you also, although you said you do accompany them, you could have visitors, that sort of thing. And one of
Yeah.
the things that I've seen is the best practice that's really easy to do is
Mhm.
on your plans and your maps, just identify an evacuation assistance area. That's a place where everyone would go on if they're on a certain floor, if they can't go downstairs. And then the fire department would know to go to one area to assist them out of the building. Does that make
Excellent.
sense?
Yeah, I agree. All excellent points, and we will take you up on that for sure.
I appreciate that.
Yeah.
And you list out the fire department, the police department. I'm fairly new, so that probably wouldn't be why it would be listed. But if you guys wanted to, you could also list the Somerville Office of Emergency Management, as well as the Somerville Local Emergency Planning Committee, which handles typically tier 2 chemicals, but would have to do with any hazardous waste and that sort of thing. It's a great resource that we would welcome you to be involved in as well. So again, the Office of Emergency Management, which I work for, and then I'm the chair of the Somerville Local Emergency Planning Committee.
Great.
Let's see. I think I got like one more thing here. Let's see.
We would enjoy working with
By
you.
the way, why I'm looking, I do want to say, very impressed with the plans, very comprehensive. A lot of them I see are a lot more succinct and don't really cover the things that we want to talk about. So that was really good. I think maybe that was it. Let's see. Oh, last thing. So you guys list training, which is awesome. Do you guys do any drills or exercises?
In terms of eye safety wash drills and, fire suppression drills and things like that?
Yeah, yeah.
Yeah, we do.
Anything to practice the stuff in the plan. Awesome. Great.
Mhm.
Well, thank you very much. I appreciate it. And I really, I mean, it's way over my head, but I like the presentation and it sounds like extremely important work. So thank you.
Cool. Yeah, we hope that we can deliver help to many patients across many diseases in the near future.
That'll be
Thank you, Bill. I appreciate the questions and suggestions. Very important. This is a relationship where we provide each other updates and advice. I did have one question concerning training frequency. It said there's 11 staff members, so I know it's relatively small, but how often are staff members receiving training? Is there introductory? Are there, biannual trainings every year?
Yeah, so I can speak on that. We have introductory training and we have an annual refresher. Basically, if they've been an employee for a year, they have an annual refresher. We also have bloodborne pathogens training, which is required, obviously. And eventually, we plan to implement some more specialized niche training when the time comes. But as far as right now, we have the introductory training for any new employee, and then the standardized annual refresher training and the bloodborne pathogen training are the three like routine trainings that are required by employees.
Excellent. Sounds pretty standard, but also it seems like there's some added layers of training there. So that's good to hear. My last question was, you had mentioned the IBC is meeting soon. I wanted to clarify that there is a requirement that there has to be 2 community members as part of that IBC. And would we be able to receive a list of names of members of the IBC? Is that possible?
Yes, I would be happy to provide those. As we have 2 tentative ones, and we haven't scheduled it yet. Just trying to figure out their schedules and giving them first dibs on times, because they're the harder members to secure their time. And I can email them to you, Colin, as soon as I have them confirmed.
Excellent. Thank you, Sarah. And if you ever need help finding IBC members, we have plenty of community folks who are interested in serving on IBCs throughout the city. I'd be more than happy to forward their information to you
Thank
for
you very
outreach.
much. I might take you up on that.
Oh,
Of
great.
course.
Yeah, that's awesome.
Thank you.
Any other questions from committee members? Rachel, go ahead.
Colin, I have a procedural question for you mostly. Without those folks named, does that change how we need to vote? Like, do we need to say pending those folks being named, or is there anything?
Yeah, we can move forward with a vote on approval and completion, but we will need to see those updates to the application to consider the license active, so we can move forward with that, and they can move forward with the process of payment and payment receivables. We can do that. That's fine.
And then just generally, so I'm Rachel Nedcarney, I'm the Director of Economic Development. We're always thrilled to hear about companies that are choosing to move to Somerville. And we hope that the building works out well for you and that there's a pathway to growth as you develop all these amazing new tools for fighting diseases.
Awesome. Yeah, I mean, so far Boynton Yards has been a great space, right? So we really enjoy it.
Excellent. Yeah, there's a lot of activity over there in Boynton Yards, so I'm happy to have it and have folks doing the work they're doing. Any other questions, comments, or concerns? All right, seeing none. I would like to move to vote for conditions of approval and completion of this application, pending some of the requests made today and then the resubmission. What that means is essentially you can move forward with payment and everything if voted on properly, and then you can receive your license as a condition that you provide that additional information and updates being requested during this meeting. Do I have a second to move forward with the vote?
Second.
Seeing a second, we will move forward. I'm sorry, my view got all messed up. What is this? Gallery. Apologies, everyone. It went full screen on me. All right. So, what is going on with this? Sorry, everyone. There it goes. Much better. Because I'm recording, you need to see everyone's faces. And anyway, I will move forward and then we will mark these down. All right. Rachel Nedcarney.
Yes.
Deputy Chief Bill Hallahan.
Yes.
Francine Rogers.
Yes.
Michael Swickus.
Yes, welcome to Somerville.
Thank you.
Bill Fisher.
Yes.
Karen Carroll.
Yes.
Colin Ziegler. Yes. And with 1, 2, 3, 4, 5, 6, 7 yeses, this is going to move forward as approved and complete on conditions that submitted documentation be updated. The next steps will be, this is going to be approved and waiting for payment. Empress can go ahead and move forward with finishing the payment through the portal, and then I'll be notified to release your license at that time. I do request that documents being requested and updates be submitted in a timely manner, probably along the same time that payment is also being received. If it needs to be a little bit later, that's fine. That's discretion that we can have there. But welcome to Somerville, and thank you all for making the time. I know we had a little bit of a scheduling kerfuffle last week, and that is on my part. So I appreciate everyone being extremely flexible with their time. If we have any other questions, comments, or concerns, feel free to raise your hand. Go ahead. All right. Seeing none, I would like to move to adjourn the meeting. Seeing any seconds. All right. And seeing a second, we will move forward with adjourning the meeting. Rachel Nedcarney.
Yes.
Francine Rogers.
Yes.
Deputy Chief Bill Hallahan.
Yes.
Michael Swickus.
Yes.
Bill Fisher.
Yes.
Karen Carroll.
Yes.
And Colin Ziegler. Thank you again for all your time. Enjoy the rest of your evening, and feel free to reach out with any questions or comments. Okay, everyone?
All right.
Have a good night, everyone.
Thank you so much, everyone.
You.
Appreciate the time.
Thanks,
Yep.
Colin. Welcome, guys.
Thank you all.